Okay, can you hear me and see my slides?
Yes.
Okay, thank you. All right, thank you very much. So we're talking about this scientific game we're developing here at the University of New Mexico that does molecular docking. So a little bit of background on the science. So we do some biomolecular modeling because they are essential for these biological processes that happen in the cell. The systems we model are receptor proteins that live on the surface of cells, and when they interact with certain ligands, they can start a process in the cell that is due to this ligand receptor complex that is formed. So the ligand finds the receptor by, so there is an energy interaction between them, and also the ligand has to fit in a certain cavity in the receptor. So the reason to do the simulations is that they help wet lab experiments to try to understand this phenomena. And having actually faster and more accurate simulations would help more rapid drug and vaccine development. So the main challenge with this type of question is that these processes, they are driven by physics in a very messy environment that is wet, there's water, there's temperature, and it's very crowded. So at the molecular level, what does this mean? So you have here this image is a receptor protein, but now I'm showing all the atoms in it and even some water molecules around. So as you can see, there are too many atoms, so it's a very crowded system. There's chemistry going on that even like the state-of-the-art software can't even capture very well the chemistry that's happening in the systems. So water itself is complicated, has a high dipole moment and it screens interactions, and high temperature that induces noise, and you have all these possible solutions. You have to run this as a stochastic simulation. So this is to say this is a very complicated system, but people have been working on this problem for a long time. And the software that is developed for molecular docking basically look at only these ligand receptor interactions without considerations of water or even motion of the atoms. Sometimes it's easier to look at the systems when they are rigid. And again, so this docking is important for cellular processes such as immune system response and metabolic processes. And if we can predict these processes, we can help finding new drugs or understand allergies, etc. So how do we actually predict this? So what do these programs do? So there are two questions. As I mentioned in the beginning, there is the interaction energy between these molecules, and the shape of the molecules also matter. So these docked positions here, so you have the receptor protein out here in beige, and then you have this blue receptor, the blue ligand. So this position here is a low potential energy. So things tend to go to low energy in physics. So, and the energy is typically negative. And the other question is, does it fit? Can you find this ligand that will fit in a cavity correctly? So it's kind of like you can imagine it as a 3D Tetris game. And the existing methods for molecular docking, you have automated docking with AutoDock, which is the most popular program used for this. They do stochastic search algorithms to find the docked positions. And you have also interactive docking, where you use player intuition to try to find the docked positions. All of these methods can also be refined with molecular dynamics. As I said in the beginning, these systems are complicated and dynamic, and after you find a solution, you can relax them with molecular dynamics. So, as I mentioned, interactive molecular docking is like a game that leverages human intuition to try to find those docked positions. So it's still challenging. It's due to the many degrees of freedom of the problem, but the user has some guidance. So there is some interface feedback, like typically as a score. also can have some haptic feedback if there is a force, so you can actually feel the forces between the molecules. And if you happen to get the ligand and the receptor colliding with each other, you can feel a repulsion force with the haptic device. And surface coloring. So you can add information to the player in the sense of like what are the physical properties of this molecule on the as a color to the surface. And this has not been tested yet. So this is what we're trying to do in this work, testing whether surface coloring, giving that information to the user, does that make them find better solutions, or how do they explore the interaction between the ligand and the receptor if they have that extra information. So our game, we call it Docket. We for this experiment, we use a desktop version with an Xbox controller. We had two molecules, both from the human immune system. We'll call them by their structured name is 3H9S for human MHC, and humanophyllin is 1FKF. The Xbox controller had vibrational feedback for collisions. So collisions don't really happen in real life. Obviously, two molecules cannot go through each other. But in the game, we allow that so that people can refine their position as they are binding to the molecule. And then they can see a score. So that there is a score that's calculated based on the energies. So how do we calculate the energies in first place? We have two terms. The first one is the van der Waals term. It goes as 1 over r to the 12 and 1 over r to the 6. It's the Lennard-Jones equation here. And r in this equation is the distance between atoms from the ligand and the receptor. So the ligand and the receptor are both kept rigid, so internal energies are not included. Only intermolecular energies are included in the calculation, so energies between the ligand and the receptor. And this energy term here is added to the electrostatic term. So this would be our total energy will be given by these, what we call the non-bonded terms in molecular biology. And the electrostatic also decays with as a power of r, 1 over r, and also has the charges of the molecule here considered. So this energy term, as you can see, if it's falling with 1 over r, when r is very small, it's close to 0, then it tends to explode. So it goes very quickly to infinity. So when you have a collision, you're pretty much overlapping these molecules. And that means that these energy values are going to be very high. And there is a region very close to the surface of the molecule when these energies can actually be low and negative. And then as you go away, since it decays with distance, these energies will go to 0 eventually. So the score is calculated from this energy. And what we want is a negative energy. The negative energy that means the low energy state for the binding. And whenever these energies are calculated to be negative, the score is calculated in this way, which transforms this negative energy that the user might not know what it means. Like if you have known scientists playing the game, they might not understand what the energy value means, but they can understand the score, and the higher score, the better. So we transform these negative energies into some number that their goal is to get the highest score possible. So with electrostatic surface coloring, what we're doing is, in addition to showing the molecules to the user, we're telling them, well, if you see these different colors, so positive charge in red and negative charge in green, in blue, that means that you can use the properties of electrostatics, meaning that positive and negative charges attract. So if your ligand is mostly blue, you're going to look for a region on the receptor that's mostly red. And like charge, they repel. So you would try not to explore, like if the ligand is mostly blue, you would try not to explore the most blue regions on the surface. So the study involved, we had 36 people come play the game, and 18 were assigned to each of the color conditions. So we had the multicolor, the MC, that we're calling the electrostatic coloring. And this, when the ligand and receptor are shown in simple colors, so just beige and blue, that we'll call that the SC, or the single color environment. And here we have 4 conditions for the 2 molecules we tested. So the 1FKF is on top here in the both color conditions, and 3H9S is in the bottom in both color conditions. So the data we collect, so for each player, we here just want to summarize this quickly, that what we observed is that for the multicolor conditions, the total number of points was significantly higher. However, there were more outliers. So these players in the multicolor condition contributed more points, but there were a few players that contributed a lot. So after we collect the data, well, what is the data we're collecting? So we're collecting the ligand locations, the XYZ locations of the ligand, the orientation, so how the player is rotating the ligand. So remember, if it is rigid, you can rotate it around an origin, which is the center of mass of the ligand. There is an interaction energy that's calculated the entire time, and the distance to the known binding site. So we know what it is, and we can tell, the user doesn't know where the binding site is, but we can tell how close or far they are. So here's an example of gameplay. We have the user starting at some position, and as they get closer, and here they get to a more negative energy, so the score starts adding up quickly, and then they collide, and then they try to correct that and rotate the molecule a little bit. So here is, once we look at the data more carefully, we find that there is a large energy distribution, so which we can pretty much split into 3 categories. So where you have these high energy states that happen whenever you have a collision. You have these negative energy states when you're close to the surface, typically, and the states with positive and low energy that could be like in between energy states. So the idea is, how can we split this energy data into 3 different states in a specific way, not just deciding where to draw these bins arbitrarily. So we use classification algorithms to classify the states. So we use dbScan to remove the outliers of the energy data. And dbScan works by connecting all points within some radius. So, and as you change this radius, more points will be connected. Here we have a plot of the largest connected component relative size as a function of this radius, and for each of the 2 proteins. So as you increase the radius, more and more points, data points get clustered. At some point, you stop growing the cluster. So the points that were not added to the cluster will be considered outliers, and we remove them from our state classification to find out where to draw the divisions between the states. So once we do that, the remaining points are used to classify the 3 energy states with k-means. So k-means will divide the data into these 3 groups based on the values of the energy. And now we have 3 energy states that the protein can be in, so that the system can be in. And we're going to call them collision states for the higher energy states, the attraction states for the lower energy states, and the neutral states for the in-between. And we want to characterize this system with a Markov model where you can go from one state to another with some transition rate. And the idea is that can these transition probabilities characterize how players explore these systems? So if you find some significant differences between these transition probabilities, that could be an indication that the player is exploring the system in electrostatic color differently than players that explore it with just simple colors. So here's the same video I played before, but now I want to show it with the Markov state system, where we will start at a neutral state, and you can see how this player will move from one state to the other as the game progresses. So far away, they're at a neutral state. At some point, when they start collecting points there, they move into attraction state. And then when they collide, they will go into collision state and back to an attraction state. And you can stay at a state for, even though you're changing your position, collecting different data points, you can stay at the same state for a while. So calculating transition probabilities. Now that we know the states and we have all the data points, we can count how many times you go from one state to another. And the transition probability is simply the ratio between this count. So if you're going from collision state to attraction state, you can count how many times you do that and divide it by all transitions from collision state. That will give you the transition probability. So here is our count for the 1FKF model for both conditions, multicolor and single color. And we have similar count for the 3H9S. And we found something something interesting here was that to go from the attraction state to the collision state in the multicolor environment, this is happening about 82% of the time. And that is significantly reduced in the single color when it's only happening about 70% of the time. So, and then also from the neutral state to the collision state, you're also having about 82% here, and that's getting reduced to 68% for the single color. We see similar trends in both molecules, which kind of gives them an indication that it's not due to the molecule geometry itself, but actually the color may be making a difference here. So all these probabilities were significant, statistically significant between the two conditions for each molecule. So we are concluding from this that these transition probabilities are in a way characterizing how players explore these different states and these different environments, and they are doing it in different ways. One thing we can also look at is the time between transitions, because we have timestamps on these data points, and we can assume now a continuous time system. So we look at the distribution of times to reach the attracting state. So whenever you are in either of the collision state or the neutral state, how long does it take to get to the attracting state? So we can look at that in elapsed time in seconds and do a histogram and fit that data to an exponential distribution. So here's the PDF. And we actually didn't find any statistical significant, any differences between these two systems. They were significant. And looking closely at the mean time to reach this attracting state, we can actually see here that it's the mean time for the single color and the multicolor are pretty much the same for this molecule 3H9S, with one outlier here for this single color. But 1FKF also shows no significant difference, but you see this the distribution for multicolor is slightly narrower than for the single color distribution. So in summary, the impact of electrostatic coloring showed that there were significant differences in just looking at transition probabilities, but not in time to reach the attracting state, which is what we really want to get to. It may have some, adding electrostatic coloring may have some impact on how long people play the game, since we got more data points from that, although there were a few outliers. But it might not help reaching the attracting state faster. So our goal from continuing this study is to look at different versions of the game. So we have a mobile version of this game, and we can test that with more players with different player characteristics and backgrounds to see if there was if the significant differences hold in these environments. But I think the take-home message here is that these Markov models can be used in these games that have well-defined states between conditions or positions on the screen that could be in principle be used to evaluate user performance. So with that, I want to thank you all for listening, and our funding agencies, and I'm happy to answer your questions. Thank you.